Hello everyone, am I audible now? So a very good evening to everyone. Okay, right, so I'm getting your messages now. So shall we start this unit 3? I hope you have done your revision by this time, because as we discuss the question, before I give the answer, you're going to try it out and then we'll discuss, okay? But we have to be a bit quick. So we'll be discussing some 10 questions in this session from the entire unit 3. Okay, Rakesh, Rajan, this is my first class. Okay, doesn't matter. So let's start, right? So the first question that I have for you is, a plasmid with linking number LK of 200, topological winding TW, number of 200, and a riding number of 0 was transformed into E. coli. The plasmid was re-isolated from the culture of the transformant. The re-isolated plasmid was found to possess a riding number of -5. Following statements are made about this observation. So this is a question from Unit 3, Part A, that is replication, where you have to have a knowledge about DNA topology in order to understand the functionality of topoisomerase. And as you all know, in case of your prokaryotic replication, topoisomerase, which is your gyrase, plays a very important role, right? So what we know, I'll just take my pen. In the meantime, all of you try it. Those who already know the formula, those who have attended the classes. So we know that linking number is equals to twist plus right. Right? Now, initially, as per the question, it is 200 equals to 200 plus 0. Okay, this was the condition of the DNA initially. Now what happened when the plasmid was re-isolated from the culture of the transformant? The re-isolated plasmid was found to possess a riding number of -5. That means what has changed? Riding number was 0 initially, now it has turned into -5, right? So that means if I want to keep my linking number as 200, my riding number is already given as -5. So obviously the twist number would change. Isn't it? So the twist number will change in order to adjust here, or it can be that the twist was 200, right? -5, so my linking number becomes 195. Okay, so let's analyze the statements. So what is the first statement? Linking of number of the re-isolated plasmid would be 195, which is justified. Yes, it is true. Linking number of the re-isolated plasmid would be 200. No, it is not correct, because your riding number is changing. So if I balance this equation, then if my riding number has become -5, then my twist 200 minus 5 linking number will become 195. Okay. Now twist of the re-isolated plasmid would remain 200. Okay, so twist is remaining 200, linking number has become -5. So therefore my re-isolated plasmid linking number has become 195. So this is justified. So the correct statements will be A and C. Alright. Now see why this is not correct. Many of you will do this, which I've already shown you. But this is not correct. Why it is not correct? Because the equation is saying that it is twist plus right. Correct? LK is equals to twist plus right. Now suppose I put right as -5. Okay? And I keep the twist as 200, then obviously this is coming as 195. Okay? But if I want to keep this linking number as 200, then will I be able to adjust here? Can I adjust here? If I keep this as 195, let me put this as 195. Plus minus is minus, right? -5. This will become 190, which is not tallying. This entire thing is not tallying. Okay, so this is wrong. This approach is wrong. This approach is wrong. Many of you will make a mistake here. Okay, this is correct. I hope you have understood. Moving to the next one. A researcher wanted to identify the enhancer sequence of a newly discovered gene. Shown below are the relevant regions of some of the reporter constructs the researcher designed to identify the enhancer. So which of the above construct can be used to identify the enhancer? Look at the construct very carefully. This is a question from Unit 3 Part B, that is your transcription, where you will come across the regulatory proteins like regulatory sequences like the enhancers, the proximal control elements, the core promoter site, the silencer. These are the regulatory sequences. Enhancer is one of the sequence where the protein where transcription factors binds, okay, or in some books they will say that activators are binding, okay. That is anything a positive regulator, a positive regulatory protein is binding here, okay. Now the details of this, why transcription factor, what kind of transcription factor, and why activators, this is something which I have already discussed in the class. For those who have attended the class, you will know about it. So for the timing, I'm just using the word positive regulatory protein, okay, which is binding. An enhancer has got certain properties, right? So now if you look at the three construct, you will see that when you have a promoter here and a reporter just after the promoter. Now, what is the function of a reporter gene? It gives you it gives you some signal or some products by which you understand that yes, this particular gene has been transcribed, right? That is reporting. It reports that yes, this gene has been activated or it has been transcribed. So obviously you need to have a promoter before that, okay. Promoter sequence is a place where your transcription factors are going to bring the RNA polymerase, right? And RNA polymerase is going to begin with the transcription. And enhancer, as we know, it activates the transcription. Okay, so this construct can be accepted. Okay, this kind of a construct can identify the functionality of the enhancer. What about the second one? Reporter gene is only present, but there is no promoter. Who will transcribe the reporter gene? If there is no promoter, polymerase will not be able to sit, right? So this construct is not correct. What about this one? See, you have a SA and then a reporter. SA is a splice site acceptor. Now, splice site acceptor is not a place where your polymerase is going to sit, right? This is a place where your splicing will take place. and splicing will splice out the reporter gene, right? So therefore, here both option A and C is correct. Why both option A and C is correct? A, we have already understood why it is correct, right? Now C is correct because, as I told you, you have a reporter gene here. Okay, so if this is spliced out, then obviously we will not get any kind of a result. Okay, so this can also give you an idea about the functionality of the enhancer. Okay, now moving on to the next one. In all organisms, it is critical that replication initiation be tightly controlled to ensure that chromosome number and the cell number remain appropriately balanced. Given below are several statements regarding regulation of replication E. coli. So, what are the statements? Hemimethylation and sequestration of Orice C by protein called SEC-A prevents initiation of replication. Availability of DNA-A protein is an important requirement for initiation of replication. The ratio of ADP to ATP is important as a high level of ADP is required for initiation of replication. Recruitment of HDA protein by sliding clamp inhibits ATP hydrolysis required for initiation of replication. Which of the above statements are not true? So you have to know about how replication is controlled, because as we all know that replication is only happening in the S phase, right? And we only restrict replication in the S phase, and we only have one replication in one cell division. Okay, so the correct answer here is C and D. Okay, hemimethylation and sequestering of Orice C by a protein called SEC-A prevents initiation of replication. This is true. Availability of DNA-A protein is important requirement for the initiation of replication. This is also true because DNA-A binds to the promoter sequence, right? And it creates a small separation of the two strands, all right, which is recognized by the helicase. Helicase simply extends that separation. Okay, so A and B is absolutely true. Now, if SEC-A protein is bound to the origin of replication, then obviously DNA-A protein will not be able to bind. Okay, so these two are true. The ratio of ADP to ATP is important as high level of ADP is required for initiation. This is not true because DNA-A proteins require ATP, not ADP. Okay. Recruitment of HDA protein by sliding clamp inhibits ATP hydrolysis required for initiation. This is also not true. Okay. Now, moving to question number 4. Are you being able to relate? Alright. Which one of the following statements related to transcription and processing of mRNA is incorrect? Read the question and tell me what should be the answer. This is again a question from your transcription and post-transcriptional modification. Any answers? Okay, I'll give you the answer. During prokaryotic transcription, DNA binding protein of RNA polymerase are altered by sigma factor. Is it so? Yes, it is. Because without sigma factor, the hollow enzyme cannot bind. Remember, you have the alpha, beta prime, right? Those factors cannot bind to the origin or the promoter site until and unless sigma factors bind. Okay, so this is a correct statement. In eukaryotic transcription, synthesis of rRNA, mRNA, and some small RNAs occurs by polymerase 1. So, polymerase 1 is for rRNA, which is true. The 18S rRNA, all the other rRNAs are synthesized by polymerase 1. Polymerase 2 is synthesizing mRNA and snRNAs also, but some of the small RNAs are synthesized by polymerase 3 as well. Okay, so this is correct. This statement is correct. Now, splicing observed in tRNA involves successive or sequential cleavage and ligation reaction, while pre-mRNA splicing proceeds through a lariat formation. It's absolutely true. If you remember the picture, you will see that in mRNA splicing there was a lariat formation, okay, which is very distinctive here. But in your tRNA splicing, it was mostly some modifications, removal of some bases, changing of the bases, removal of some extra sequences. That's it, right? So this statement is also correct. Now, if you go into the fourth statement, mRNA with premature stop codons are degraded by nonsense mediated decay, and mRNAs without an in-frame stop codon get accumulated and translated in the cytoplasm. This statement is incorrect. Now you must be thinking, what is this nonsense mediated decay and all? Okay, this is actually a surveillance pathway which is present in all the eukaryotes. And this pathway mainly works to reduce errors in the gene expression by eliminating all the mRNA transcripts which contain the premature stop codons. Okay, because obviously, if these mRNAs gets translated, then it will lead to deleterious gain of function mutations, or maybe dominant negative activity of the proteins, right? So when your non-saturated decay activity is on, it will not allow any in-frame stop codon to get accumulated and translated. Okay, so this statement is wrong. Okay, moving on to the next statement. Next question. In an in vitro experiment using radio-labeled nucleotides, a researcher is trying to analyze a possible products or DNA replication by resolving the products using urea polyacrylamide gel electrophoresis. In one experimental setup, RNase H was added, that is set 1, while in another set, no RNase H was added, that is set 2. The possible observation of this experiment could be what? So who is going to give me the answer? Okay, Malini has tried. Anyone else? So see, when you have a higher percentage of this polyacrylamide, no, it resolves the lower molecular weight fragments. So the combination of urea and the high temperature, like temperature of 45 to 55 degrees centigrade during the gel run, will only allow the separation of unstructured DNA or the RNA molecules. All right. So the correct answer here is option 2. So they are saying that there is no difference in the mobility of labeled DNA fragments between set 1 and set 2. No, there will be a difference. Why is it so? Because in one case there is RNase H added, in the other there is no RNase H added. Now just think, what is the function of RNase H? RNase H being an nuclease, it is going to remove the primer sequences. In your eukaryotic replication, the primers are removed mainly in the Okazaki fragment. There are a lot of these primers, right? So the primers are removed by RNase H, and in the place of RNA, in the case of your primer, the DNTPs are added to complete the DNA. Right? So there is a distinct difference in the mobility of newly synthesized labeled DNA fragments between your set 1 and set 2. Okay? Now the mobility of the newly synthesized labeled DNA in case of set 1 is faster as compared to set 2. Which is also true, because in the case of your set 2, you're not adding your RNase H, right? So you have DNTPs and RNTPs. So the structure, the mobility will be different. Whereas in the case of set 1, you just have the DNTPs. No RNTP is there. Alright. So in the case of the first one, it is faster as compared to the set 2. So that means the statement D is wrong. Okay, so the correct statement here is B and C. Yeah, okay. So read the question. Read the question very carefully. That is very, very important. So far, we have discussed 5 questions. What do you think? Are the questions very difficult? Do you think the questions are very difficult? Like, even if you don't know a particular word, like in the previous one, in question number 4, you might not know what is nonsense mediated decay. Okay, maybe this is new to you, but the other statements, did you notice that these are something that you must have studied, or you should have studied while you are studying your transcription? These are very general statements, very general information. So with the general information, you can straight away rule out statement 1, 2 and 3. So that wouldn't make it difficult to say that statement 4 is incorrect. Okay, so these are the small tricks that you need to apply when you are reading the questions. Yes or no? I'm not getting any answers from you. Did you find this very difficult? No, right? Because mostly so far, whatever we've studied, it is absolutely your subject based. When you are talking about replication transcription factors, not only the process, but the regulation of the process, the functionality of the enzymes, they are very important, right? And this is what we have already discussed in the class as well, if you recollect. Okay, just a small announcement that the Josh batch will be starting soon. All right, for your December CSIR NET December 2024 and June 2025 exam. Uh, you will get special supplements on general aptitude and practice papers, AIM NET test series, 24 into 7 chat support for academic doubt resolution, free access to Biotechnica's online study portal, free access to Biotechnica Concept eCloud, which is important for your revision purposes. Free internships also you will get, okay, which will enhance your career, your prospect towards your PhD, your research. You will get some mind maps for quick revision purposes, self-study booster package, study materials, AI-powered AIMNet e-test series, tough question bank. So you're getting a lot of these test series apart from the weekly test and the quest that we conduct. You're getting a lot of test questions to practice and practice, because practice makes a man perfect. Apart from that, concept wheel, concept cloud, which is definitely going to help you revise the topics, okay? And double success guarantee is there because we take the classes, everyone takes the classes very sincerely here, and we try to resolve all your queries, and premium guidance via chat support and live sessions from the best teachers you will get. Okay, so the batch starts from the 1st of July. So if you are serious about your exam, you can think about it, right? Now let's get back to the question answer discussion once again. So the question number 6. The marrow-diploid strain of E. coli with the genotype F plus OCZ minus Y plus O plus Z plus Y plus was constructed. The activity of beta galactosidase enzyme was measured in this strain upon following treatments. What treatments? No induction, first treatment. Second is induction with N moles of IPTG, induction with N moles of lactose, induction with N moles of lactose in presence of N moles of glucose. Which of the following graphs depict the expected trends in beta galactosidase activity under the 4 different conditions? Now, this is from your lac operon, right? O is for the operator, Z is for the beta galactosidase, Y is for your permease, right? OC means it is a constitutive operator. That means the operator is always functional. Okay. Z minus means there is no beta galactosidase. Y plus is there. That means permease is present. And this is a marrow diploid condition. So you have O plus, Z plus, Y plus. Okay. Now, IPTG, galactose, glucose, these are what? These are all your inducers, which goes and binds with the repressor. Okay. So just think about it. I'll give you time. Think what should be the outcome. OC, Z minus Y plus. Okay, O plus Z plus Y plus. Now you have to know which is cis-acting, which is trans-acting, in order to understand how to analyze these merrodiploid situations. Okay, Malini is saying option 2. I'm waiting for others also. I'm giving you time. Try it yourself. This is very interesting. So take every situation. First, you analyze a general situation. Okay. First, analyze a general situation. Understand you have to find out the beta galactosidase activity. Okay. That means you have to find out the functionality of the Z gene. So under which conditions the Z will be active, it will be inactive. That you have to analyze. Okay. So I'm getting mixed answers. Everyone try, whether it is right or wrong, try it, because the more you try, that means you are practicing. The correct answer here is option 3. Now I'll just tell you the graphs, the numbering, if it's not clear. This is 1, this is 2, this is 3, this is 4. Okay, so let us understand. First, there is no induction. Okay, so when there is no induction, that means what? Look, okay, first let us look at this merrodiploid condition. Here, even though the operator is constitutive, that means the repressor won't be able to bind here. You don't have the beta galactosidase gene, right? So you're not going to get anything out of this particular strain here, this particular construct. Whatever you're going to get is from here, which is the Z plus, okay? Because Z operator is cis-acting, it's not trans-acting. This operator is normal, okay? Plus means it is a normal one. It cannot go and, activate anything over here. Moreover, there is no Z gene present. So you have to look into this construct. Now, if this is a normal operator, that means it will bind to your repressor. Then the inducer has to come and bind to the repressor, take it away from the operator. Only then the polymerase will be able to bind to the operator and transcribe the genes. Right? So initially, when there is no inducer, no induction, matlab there is no inducer to remove the repressor. Okay. Then what is happening? Repressor is bound to your operator. Okay, so many of you will say that it will be 0. The activity of beta galactosidase will be 0, and you must be thinking that option 2 is the correct answer. But no, it is option 3. Here is a trick. Why? Because understand that repressor, they don't always sit on the operator. Okay, even without an induction, they will leave the operator side and then again they will come back and bind to it. Okay, now in this small time, in this very short time, okay, you will get a little bit of beta galactosidase expression. What we call it as a leaky expression. All right, so you will get a small amount of beta galactosidase expression. It will not be absolutely 0. This is wrong. Okay, so this makes the graph 2 wrong. Okay, and graph 1 accordingly is also wrong, because you will not get such high expression of beta galactosidase. It is a leaky expression. Okay, so tiny bit of expression you will get. Now, next one, induction with n moles of IPTG. IPTG is an inducer, right? So it will remove the repressor. So your operator will be free to transcribe the Z gene, right? Your polymerase will be able to bind to operator and you will get the Z gene. So you're going to get a high level of Z expression, okay, which is absolutely correct here. Now, induction with n moles of lactose. Now, lactose, the interesting part here is that even though lactose is a inducer, it gets converted to allolactose, and allolactose functions as an inducer. Lactose is also a substrate. Okay, so therefore you will definitely get a high expression here, but not as high as your IPTG, because IPTG doesn't work as a substrate, it only works as your inducer. Okay, so the moles of IPTG will not reduce. Whatever amount of moles you will add initially, it will remain as it is. But the moles of lactose will start depleting, because lactose will be used up as a substrate as well. Okay, so slightly less expression. In the next one, induction of n moles of lactose in the presence of n moles of glucose. Now see, if glucose is present, lactose doesn't work. Okay, so glucose will be basically broken down. Only after glucose is broken down, then the lactose will be broken down, then the operon will be initiated. Okay, so you will get an expression, but a very little expression you will get. Okay, so this is the thing. IPTG is a gratuitous inducer. Okay, so maximum amount of expression you will get from your IPTG. I hope this is clear to everyone. Okay, moving on to question number 7. So the following statements are made with respect to merodiploids in the lac operon, where I is the lac repressor, O is the lac operator, Z is the lac Z gene encoding beta galactosidase, and Y is the lac Y gene encoding permease. So they have given you certain construct, and now you have to find out the correct combination. So see here, they have given you the induced, the repressor. Okay, I is negative here. So there is a error in writing. It is I negative O plus. Read it like this: Z plus Y minus. Okay, this is not I minus O plus Z plus Y. No, it is not like that. It is, it is showing you the state of the gene. So these are in the superscript. So the Z is inducible and likewise is constitutively expressed. Does it mean so? Try it out, everyone. Any answers? Analyze each and every statement. Remember your lac I. This is transacting. I is transacting. That means in a merodiploid condition, I can work on both the promoters. I mean both the operators. Okay, you have tried Malini at least. Okay, Vijay Lakshmi also. What about the others? In this entire session, I find Malini and Vijay Lakshmi is only answering, and sometimes Alka has answered. Try. So the correct statement here is option 3. The correct answer is option 3. Okay, you have to find the combination. So let's see what they are saying. So in the first case, I'll write it down. I plus O constitutive Z minus Y plus, right? Now see, here is I minus, that means inducer is, repressor is not present, okay? But here you have a repressor. I plus, here you have a repressor, correct? So this repressor will be working on both the operators. It is capable of working on both the operators, okay? But here this is operator constitutive. That means repressor cannot bind to this operator, okay? It will not be able to bind here. So inducer will be binding to this operator. Now this operator has got a positive Z gene, okay? So if it is binding to positive Z gene, then what will happen? This Z, the expression of this Z will be dependent on the presence of an inducer, right? When the repressor is binding to the normal operator, okay, then the expression of Z is dependent upon the presence of a repressor. Sorry, inducer. Okay, so lag Z is inducible, correct? What about lag Y? Here there is no presence of lag Y. It is negative. But here I have a lag YG. Okay. But here the operator is constitutive. That means the repressor cannot bind to the operator. Hence, lag Y will be constitutively expressed. So this is correct. In the next case, what do we have here? They are saying that I is plus, O is plus, Z is also plus, Y is minus, and in the other strand you have I negative, O plus, Z negative, Y plus. Now if I is negative, then it is non-functional, okay? But here I have an I plus which can work on both this and this operator, and here both the operators are normal operators, okay? But here you don't have a Z gene. Here you have a Z gene, okay? So this Z will be inducible. This Z will be inducible, okay? Here you don't have a Y, but here you have a G. So this repressor can control this operator, which is going to control the Y gene. And here again, since the operator is normal, therefore it is again going to be dependent upon the presence of inducer. So Y is also going to be inducible. Okay, so both are going to be inducible. This is correct. In the next set, I have I plus O constitutive, Z plus, Y minus. Okay, and I have I minus, O plus, Z minus, and Y plus. Right? So again, see, this is non-functional. This is functional. Okay. Here again, the I will be able to control this operator because here the operator is constitutive. So let us analyze. They are saying that lag Z is constitutively expressed. So lag Z is under the expression of constitutive operator. So this is constitutively expressed, which is correct. Okay. There is no Y here, but there is a Y present here, which is under the expression of a normal operator. So therefore, Y will be inducible. This is correct. Now, if I analyze the third statement, so first three statements A, B, and C are correct. Okay, these three statements are correct. If you look at the fourth one, the last one, I negative, O plus, Z plus, Y minus. And here you have I plus, O constitutive, Z minus, Y plus. So again, see, this is non-functional, okay? This I will be able to control this operator. Under this operator, you have Z, so therefore this one will be inducible. But here they have written it is constitutively expressed. This is wrong. Z here will be inducible. Okay, Y will be constitutively expressed because Y is under OC. But here they have said it is inducible, so it is wrong. Okay, so statement D is wrong. I hope everyone is clear. So whenever you get a question like this, take up each of the combination and analyze it separately. Try to tally it with the statement. How Y and Z are both inducible in case of your B? See, I'm writing it as again A plus I plus O plus Z plus Y minus, and here you have I minus O plus Z minus Y plus, right? So see, this is non-functional, this is functional, okay? Now it is working on both the operators which are normal. Now here you have Z plus Y minus, so Y is not present here, only Z is present. So Z is inducible, okay, because this is a normal operator, so repressor will bind to the operator, inducer will come and remove the repressor, Z will be expressible. Okay, so Z is expressible only in the presence of inducer. So it is inducible. What about Y? Here you don't have Z, you have Y. So this operator is functioning on Y. Right? Same explanation, Y will only get expressed under the presence of an inducer. So therefore Y is also becoming inducible. I hope now it is clear, Alka. Okay. So if it is not clear to you, I have a picture as well. Whatever I have explained, it is there here. All right. Okay. So I'll just skip this part. Later on, if you want to go through this, you can pause the video and again go through this picture. Okay. Right now, I'm not going into it because I've already explained it. Let's move on with the question. So question number 8. Fidelity of protein synthesis depends to a large extent on the accuracy of amino acylation of tRNA with correct amino acids. However, given that the side chains of many amino acids are not sufficiently different, amino acid tRNA synthetase are often prone to misacylate the tRNA. One such example of misacylation is tRNA threonine by uh, threonine amino acid synthase. Okay. In this content, following statements are being made about E. coli, uh, threonine amino acid tRNA synthesis. So out of the statements, you have to choose the correct statements. So this is absolutely based on the amino acylation property of the amino acid tRNA synthetase, its proofreading activity and what it does. Okay, this is in the chapter of your translation. Okay, so translation is not only about studying the genetic code, the properties of it. It is not just about the factors that will initiate the translation, okay, but also about the enzymes and particularly the amino acid tRNA synthetase. So here the correct answer is statement B and C. It possesses a distinct editing site that preferentially deacylates misacylated tRNA threonine. All right, and The editing of the misacylated tRNA threonine occurs frequently in cysts before the release of misacylated tRNA threonine. Clear? These two statements are correct. Okay. Now moving on to question number 9. So question number 9 is from splicing, that is your post-transcriptional modification. The splicing occurs simultaneously with transcription. So they are saying that during maturation process of some RNA molecules, formation of a 2' 5' phosphodiester bond takes place. Following statements are made about this phenomena. So you have to select the statements which are true. That means the correct statements. So this is straight away from the theory. So what should be the correct answer? Option 3 you're saying. Catalytic spliceosome is 2,6. The correct answer is option 3. Spliceosomes mediated okay. Spliceosome mediated removal of intronic sequences occurs through the formation of 2 to 5' phosphodiester bond. 5' is the site on your 5' exon, right? That is a 5', and 2' is the internal adenine. You have internal adenine has got a 2' OH group. So that OH group makes the hydrophilic attack at the 5' splice site. Okay? So this is correct. Next one, removal of group 2 introns occurs through the formation of 2,5 phosphodiester bond. This is also correct because here also you have an internal adenine which is giving the OH group. Okay, the 2' OH group, which makes the attack at the 5' uh, exon. Now, enzymatic removal of tRNA from the removal of introns from the yeast tRNA precursors involve 2 to 5. This is wrong. Okay, our NSP mediated 5' end maturation of tRNA precursors involves formation of 2,5. No, this is also wrong. Okay, only in spliceosomal intron and in the group 2 introns you have a 2,5 phosphodiester formation. And now is the last question. The complex responses to different of DNA damage in both prokaryotes, eukaryotes fall into 3 main categories. Damage bypass, damage reversal, and damage removal. Many proteins are isolated like DNA methyltransferase, DNA glycosylase, and DNA polymerase 4. So you have to match the correct enzyme associated with the correct kind of your DNA damage. In tRNA, there is no intronic removal. Okay, there are some bases are removed, some sequences are removed. Okay, there is no such elaborate processing. Some bases are modified. Okay. Okay, bypass. Bypass, remember translation, Malini? We have talked about translation. What were you doing? You were not repairing that damage, right? But you were just allowing the polymerase to pass through that damage. Okay, Vijayalakshmi is saying option 1. Yes, exactly, Vijayalakshmi. Randomly add BP, base pairs. Okay, so the correct answer here is option 3. Okay, let us match it. DNA bypass is done by your polymerase 4. Damage reversal is done by your methyl transferase, because this is following your mismatch mismatch repair. Okay, where methylation is very important. There is a damage, there is a mismatch there, and you're editing that mismatch with the correct one. Okay, so mismatch repair is talking about. Damage removal, this is by your DNA glycosylase, base excision repair. Okay. So see here also, if you don't know exactly about damage bypass, you know about damage reversal and damage removal, isn't it? DNA methyltransferase and DNA glycosylase, you know about this, even if you have not studied about translation. So even if you can match methyltransferase and glycosylase with the correct kind of a damage, the third one automatically will be set. Yes, exactly, Ramachandran. Right. Yeah, okay. Right. So, when you're studying about these processes, no, just don't study it as a process. Okay, okay, this is happening, this is happening, this is happening. Okay. This is all about DNA repair. So, how is the damage, how you're repairing, that also should be kept in mind when you're studying. All right. So, with that, we come to the end of this discussion. I hope you have found this helpful. Was this helpful to you all? So, see, we are conducting these question answer discussions with you. If it was possible for you, I would say, go through the topics once. Okay, revise the topics once, because then you will be able to recollect the things very easily and connect to the questions very easily. Okay, so that's it from my side today. Thank you, everyone. So by now you know which are the portions that you need to revise. So go ahead with it. Okay. See you again with another series on Unit 10. That is your ecology. Thank you and bye bye. Take care.